Good morning, Doc. Good morning, classmates. We are the Group 4 and we will be presenting the blood. I am Danish Kate Balmilero together with my group mates, Miss Excel Jean Lopez, Miss Precious Buenafe, Miss Cherimar Ventura, and Miss Angela Silvestre. The goal of this report is for us to be able to name the components of human blood tissue and their functions, to know how common blood tests are performed, and state the limitation of clinical blood testing.
Blood sampling and blood smear. Blood sampling. Blood sampling for clinical testing usually requires only tiny amounts of blood, often only a few drops. Okay? Blood sampling ay yung pag-collect mo ng blood samples para matest mo yung blood. Example is yung pinag-test mo ng sugar ng blood or blood typing. Kahit few drop lang na blood ay madedetermine mo kung anong type na blood using a liquid na ipapatak mo doon sa blood. Next is blood smear. Investigate blood tissue by looking at several prepared blood smear slides. As the name implies, it is merely some blood smeared on a slide, then prepared for microscopic examination. Okay? Blood smear is yung blood na ilalagay sa slide. Yun yung time na itetest yung blood para ma-determine yung white blood cells or the RBC.
Differential white blood cells. First is leukocytes. Leukocyte is a white blood cells. It is a cellular component of the blood that lacks hemoglobin, depend from the diseases and infections by destroying cancer cells. For leukocytes have lymphocyte and monocyte, and from the granular, it is a eosinophil and basophil and neutrophil. Erythrocytes. Erythrocytes is a red blood cells contain pigment hemoglobin which imparts the red color to blood and transport oxygen and carbon dioxide from the tissues. Platelets. Platelets are fragments of cells consisting small amount of cytoplasm surrounded by a cell membrane producing red bone marrow from megakaryocytes, which plays an important role in preventing blood loss. Colorless cell fragments in our blood that forms clots and stop or preventing bleeding. Platelets is made in red bone marrow, that sponge-like tissue inside of our bones, which contain stem cells that develop into red blood cells, white blood cells, and the platelets. For the granulocytes, granulocytes is a type of white blood cells that has small granules that contain protein, helps the body fight bacterial infections. For the type of granulocytes, the first is neutrophil. Neutrophil is a pinkish granules and lobe nuclei that resemble several links of sausage. Biggest number of all kinds of the white blood cells. They kill and digest bacteria and fungi to help body fight infections and heal wounds. The second is eosinophil. Eosinophil is a red granules and two-lobe dark nuclei, which is white blood cells containing granules that are readily stained by eosin. The third one is the basophil. Basophil is a fever granules which are bluish tint of variable size, one of the several kinds of white blood cells in our body. Blood cells makes up less than 1% of all the circulating blood cells, least abundant in all the mammals. The fourth one is the lymphocytes. Lymphocytes, they are made in bone marrow and found in the blood and lymph tissues. Sometimes, lymphocytes appears to have no cytoplasm. The last one is the monocytes. Monocytes is a granular leukocytes with large variable-shaped nuclei. They are the largest type that differentiate into macrophages and conventional dendritic cells.
Hematocrit and hemoglobin determination. So in hematocrit, A plasma is a fluid matrix of blood tissue, and it has 91% of water, 7% of proteins, and 2% of other compounds such as ions, nutrients, gases, waste products, and regulatory substances. And it is also composed of plasma proteins such as albumin and antibodies. It has also other dissolved substances such as urea, oxygen, and glucose, and has formed elements such as red blood cells, white blood cells, and platelets. So let's talk about hematocrit test. What is a hematocrit test? A hematocrit test is also known as PCV or packed cell volume. It involves the packing of all the cells in the blood sample at one end of the tube. So this is the step in hematocrit test. The blood sample is placed in the centrifuge, this one as you can see in the photo. A centrifuge is a machine that spins a tube of blood and allows centrifugal force to push the heavy blood cells to the bottom. And once the cells are packed, the proportion of cells to total volume can be determined. This process is also used to determine whether it has a shortage of red blood cell that may cause anemia, or it has an excess red blood cells that may cause polycythemia and dehydration. So, this is the procedure of the hematocrit test. First, you need to fill a heparinized capillary tube. Second, seal the unused end of the tube and place the tube in one of the numbered grooves of the microhematocrit centrifuge. Spin the tube for about 4 minutes and then remove the tube from centrifuge and measure the red blood cells and the plasma. Then divide the length of RBC or red blood cells section by the total of sample length times 100 to get the percent volume of packed red blood cells. So, this is the normal hematocrit. For the male, it is 40% to 54%, and the female is 38% to 47%. The amount of hematocrit increase. If increase, it can cause severe dehydration, shock, and polycythemia. And if it is decreased, it can cause anemia, leukemia, cirrhosis, and hyperthyroidism. Next is the hemoglobin. Hemoglobin is known as the pigment inside red blood cells, and one of its functions is to transport blood gases. And also hemoglobin picks up oxygen in the lungs and releases oxygen in other tissues. And please take note that a denser color of sample has the higher content of hemoglobin pigment. In hemoglobin determination, the more accurate methods use colorimeters and spectrophotometers that measures the percentage of filtered light transmitted through the sample. And another accurate instrument is the hemoglobinometer, while the Talkis method is less accurate. So here's the procedure of the Talkis method. First, place a drop of blood on the special paper in the Talkis. The red blood cell break open in the paper. So this one. Then, allow the drop to dry long enough to lose the glossy appearance but not long enough for it to become brown. And Compare the color of the blood spot to the colors in the Talkis chart. And record the result of HB per 100 ml of blood. Then determine the HB content by matching the colors. So here's the normal hemoglobin for male. 14 to 16.5 grams. And for the female, it should be 12 to 15 grams. If the hemoglobin increase, it can cause polycythemia, congestive heart failure, obstructive pulmonary disease, and also it can affect the high altitudes. If it decreases, it can cause anemia, hyperthyroidism, liver cirrhosis, and severe hemorrhage.
Good morning, Doc. So the next one is blood typing. I am Miss Lopez from BSN 1A Group 4. So surface proteins acts as an identification tags, whereas non-cell proteins are detected to destroy or inhibit foreign proteins. So cell surface proteins are hereditary. So next is tissue rejection. If foreign tissue is transplanted or blood is transfused, immune system will destroy cells that don't have self-protein. So to prevent tissue rejection, biologists must determine the blood cell marker that may elicit life-threatening immune reactions. So next is blood typing. We have antigen and antibody. Whereas antigen is a molecule that elicits an immune response, and the antibody is the plasma protein produced by the immune system. So next is the agglutination reaction, where it is a type of antigen-antibody reaction. This clumping is a reaction commonly seen when incompatible blood types are transfused. Here in agglutination reaction, antigens are glued together with antibodies like flies on fly paper. So next we have this slide, the blood type, antigen present and the antibody present. Whereas blood typing, we used an anti-sera containing antibodies against surface antigen. So if there is an antigen A present, it is blood type A. Whereas if there is antibody AB present, it is a blood type O, and so on. So we have the RH or antigen D, where it does not elicit an immune reaction in someone who does not have it until a second exposure to the antigen. RH typing is similar to ABO test, whereas person with antigen are RH positive and the person without antigen is an RH negative. So this is an example of blood typing. Anti-sera, anti-A, anti-B, and anti-D. The blue one is for anti-A, blue anti-B, and the last one is for the anti-D. So the first one is, I believe, AB plus. The next one is A positive, and then O positive, and then B positive.